Hello students, this is Dr. Faizan Mirza. We are discussing the topic of homeostasis and how the kidney is functioning in promoting the first step of urine formation, which is ultrafiltration. So we know that this is the structure of a nephron. In the nephron, this is the Bowman's capsule. This region is called as the Bowman's space. So blood comes from the afferent arteriole. This comes into the glomerulus. Glomerulus is a specialized capillary which is having fenestrations. Any structure which is smaller than the gaps, which are the fenestrations present in the glomerular wall, they pass out into the Bowman's space. This region is the Bowman's space. And the cells that make up the lining of the Bowman's capsule, they also have gaps which are called filtration slits. So this is the process of ultrafiltration which is highlighted in these orange arrows here. And the unfiltered blood then goes into the efferent arteriole. The unfiltered blood will primarily be having those substances which could not pass out into the filtrate. So how this process of filtration occurs. So if we take this diagram and we try to analyze what's happening here. So this is the first step of urine formation ultrafiltration. So this is the nephron part, Bowman's capsule. It's a cup-shaped structure, and this is a single cell lining. The Bowman's capsule is having a single cell lining, and this layer is also a single cell lining. The space between the two is called as the Bowman's space. This is the diagram of the nephron that we have already discussed previously. So this is the Bowman's space here. We know that the Bowman's capsule is having two cell linings. The one which is facing right next to the glomerulus is the visceral layer, and this one is the parietal layer, the outer layer. And the space between the visceral layer and the parietal layer is called as the Bowman's space. So if we just look at the diagram and we try to see where the glomerulus is. So the glomerulus is in close proximity to the visceral layer of the Bowman's capsule. So this is where the glomerular filtration will be occurring. So here you can see that this is the visceral lining of the Bowman's capsule. This is the epithelial cells. And this is the glomerulus here. This blood comes into the glomerulus. It flows from a larger diameter vessel, the afferent arteriole, comes into the glomerulus. The blood gets filtered out. These tiny arrows, these show how the filtration process is occurring. And the unfiltered blood goes through the narrowed vessel, which is the efferent arteriole. Both are arterioles, so they both have oxygenated blood. Efferent takes the blood away from the glomerulus. Now, if we try to just magnify this region where the visceral lining of the Bowman's capsule was coming in close proximity to the glomerular network or the capillary here. So we see this is the magnified version. This is the inside of the glomerulus. This is the red blood cell. This is the plasma. So everything dissolved in the blood plasma will be present here. These are the capillary. This is the cells that make up the capillary wall. We know that capillaries are designed for exchange. But glomerular capillaries, this network is called glomerulus because it's a different set of capillaries altogether. Because these capillary endothelial cells, this is an endothelial cell of the capillary, a cell that's always present in the capillary. But the adjacent cells, adjacent endothelial cells of this capillary will have tiny gaps. These are the circular holes. These circular holes in the endothelial lining of the capillary wall of the glomerulus are called as fenestrations. So this is the first size barrier for filtration to occur. So any substance which is dissolved in the blood plasma which is smaller than the fenestration can easily pass out from between the gaps present between the adjacent endothelial cells. This is the basement membrane. The basement membrane is a meshwork, or you can say a network of these proteins which adhere to the single cell lining, and they also provide some kind of barrier but not exactly a barrier. So the filtration just the filtrate just moves from the fenestrations into this region. Now these are the cells that line up the visceral lining of the Bowman's capsule. So this was the visceral lining of the Bowman's capsule. If we take this rounded mass here, so this is the cell. You can see this cell is having a circular region here. This is the nucleus of the cell, the cytoplasm. The endings of this cell are having podocytes. So this is a podocyte, another podocyte. So all these podocytes cells are present in the capsule wall of the visceral lining. These are the foot processes of the podocyte cell. These foot processes are extended towards the glomerular capillary. So if you look at that into a 3D image of a glomerular capillary and the podocyte cell, so you see that the podocyte extensions, these foot processes actually wrap over the glomerular capillary to minimize the distance for exchange to occur. Now the gaps are present between the podocyte cells as well, and between the foot process of the podocyte cells as well, and these gaps are called as the filtration slits. So any structure or any solute which is a smaller, any cell or any component in the plasma which is a smaller then the fenestration as well as the filtration slit will pass out into the filtrate. So it enters into the Bowman's space and then it will go ahead into the nephron. So what structures can we see here? So for example, there are ions such as sodium, potassium, magnesium, bicarbonate, H plus ion, glucose, amino acid, inorganic phosphate, vitamins. So all of them can pass out into the filtrate because they all are smaller than the fenestration. Even water will pass out here in large quantities. White blood cells can squeeze as well, but large proteins will stay back. But small proteins such as antibodies and dipeptides and tripeptides are small, you can say certain hormones which are very small, they can also pass out here. So when they pass out, so this is all the filtrate is. But what you wanted to excrete, you wanted to excrete out the metabolic waste, the creatinine, the urea and uric acid. So yes, they also pass out into the filtrate. So this is the complete set of what's present in the filtrate. Now this is the Bowman's space, and here I am labeling the Bowman's space as well. So what's present in the Bowman's space is the ultrafiltrate, and this ultrafiltrate is what eventually will form your urine. This is not the urine as yet, because there are a lot of useful substances present here which need to be reabsorbed back. So the final fluid will be the urine there. So what actually happened? Blood came from the renal artery into the efferent arteriole. It turned into the glomerulus. From the glomerulus, the blood got filtered. Filtered blood was the filtrate, and the filtrate actually moved from to the Bowman's capsule and then the Bowman's space, and then ahead into the nephron. What about the unfiltered blood? The unfiltered blood goes into the efferent arteriole. This is having primarily red blood cells, large solutes, large proteins, and even some waste which could not get filtered out. So they go into the efferent arteriole, and ahead from the efferent arteriole they go into the capillary network present across the nephron, which includes your peritubular capillaries and the vasa recta. That's it from my side. Thank you so much.